Café Scientifique is a monthly series of expert-led discussions on science and culture presented by the Bell Museum of Natural History. For more information about the Bell Museum, or to find out about upcoming Café Scientifique programs, visit bellmuseum.org or find us on Facebook and Twitter.
Okay, welcome to the Café Scientifique. Yeah! Woo! So I'm a retired U of M employee. I am now just volunteering, and so I can drink up here. Yeah, all right. At least until I get found out and get yelled at by somebody, who I'm sure somebody will. But anyways, what? Okay, good. Let's all be sworn to secrecy. Okay, who brings the Café Scientifique to the public? Can anybody answer that? It's the Bell Museum. Okay, very good. Okay, I'm turning it over to Leah and Gail. Thank you. Woohoo!
Thank you, Kevin, as always. He also was our curator of K-12 education and our honeybee keeper for more than 20 years, 30 years, and also my mentor and the person who hired me. So I'm very fond of him, and he deserves a big round of applause.
In the last few years, I kept.
What were you saying?
Last few years, I kept the HR department in business.
Indeed. All right, well, and my name is Leah Peterson, as Kevin mentioned, and I'm the Adult Programs Coordinator at the Bell Museum. Thank you, as always, for being here. We're thrilled to have you, and we have an amazing presentation lined up for you tonight. I want to turn the mic over to Dr. Gail Celio. Just a quick note about how she came to be here tonight. We met because Sri Kishan Nair, who was our last resident artist with the McKnight-funded Resident Artist Research Project, was very interested in taking pictures, photographs with his macro lens camera, blowing them up, and then manipulating them digitally into these very beautiful, large-scale, printed-on fabric creations that are in our gallery through March 5th. And one of the things that we do with the Resident Artist Research Project is that we connect our artists to our scientific resources, curators, collections, and all kinds of other outlets around the university that in areas that they're interested in, so that they can create new works with us. And then McKnight Foundation helps us pay them in a meaningful way also that's commensurate with other professionals at the university. And so Sri Kishan was really interested in seeing little worlds become big, beautiful, detailed places that you could kind of lose yourself in, and these new landscapes that could be created from super magnification. And so we went to university imaging centers, and Dr. Celio was kind enough to be our host and to talk to us about the technologies of imaging and microscopy, and she taught me to say microscopy instead of microscopy, which I still have to practice every time, thank you. But so that's how we came to connect, and she was just wonderful, and we wanted to bring her in for a cafe. So here she is. Check her out also on KFAI. She did a spot yesterday morning at 7:45 that's archived now on the Morning Blend. So give her a listen, and without further ado, let me introduce Dr. Celio, Dr. Gail Celio.
Thank you for having me. Thank you, Leah. Thank you, Kevin. My role at the University Imaging Centers, we have a wide variety of microscopes, almost 24 ways to image things so we can collect data. My particular specialty is electron microscopy, and you will definitely be seeing that here. But I wanted to show you some of the things that we can see on a very small scale, introduce you to some of the projects that we've crossed paths with, and hopefully you'll see some really cool stuff in your opinion. So yes, this is us. We have 3 sites on the on the Twin Cities campus. We have 2 sites on the East Bank at Jackson Hall and at the new CCRB building, Cancer and Cardiovascular Research Building. It still has new building smell. You should go in it just for that. And also on the St. Paul campus, which is where I work. And yes, there is a campus on St. Paul, I promise you. It's just a bus ride away. Come on over.
And the Bell Museum will be there soon.
Yes, and then you'll have to come because of the Bell Museum, absolutely. So we have many services in terms of the microscopes that we're able to use. We also have the services for preparing samples so that you can collect your data. We can collect your data for you, or we can train you how to use it yourself so you can operate it independently. So there are options. So how do we decide what to do, which instruments to use to collect the information that a client is interested in? There is a wide range, and it overlaps. So the unaided eye, we can see humans, great. Also see frog eggs. There are things that we can see with the naked eye. That's fine. Under a light microscope, we can see some of that, but also getting into the cellular level in terms of eukaryotic cells, the organelles inside, bacteria. And then there's also overlap with the electron microscopes. They can definitely see cellular features, but then you also get into smaller structures. You can see viruses, very small organelles like ribosomes. And depending on if you've got a really good electron microscope, you can get into individual protein molecules and maybe even very small molecules. There are microscopes on campus that can do that. Ours does not, I will admit. The history of the facility where I'm at has been closely tied to the College of Biological Sciences. So the majority of what you're going to be seeing are biological samples, and also because my background is in plants and fungi, you're going to see a whole bunch of spores and a whole bunch of plants, and it's going to be great. I'm not biased at all. This is a great site to see, get a sense of scale. All right, so we've got paper and a coffee bean and a grain of rice. We're taking a fantastic voyage. We've got grain of salt. We've got an amoeba proteus, a paramecium, and a human egg and the sperm compared to it. Photoreceptors from the eyes. A skin cell. There's that red blood cell. 8 microns. All right, there's a baker's yeast. That X chromosome, that's condensed and during replication, so just for size comparison. Further down, there's the E. coli bacterium, the mitochondrion, a lysosome, which is a structure, an organelle in the cell that's pretty much the trash compactor of the cell. Now we're down into viruses. And even viruses are different sizes. Okay, hepatitis, rhinovirus, ribosomes. Now we're getting into molecules. We've got proteins here, hemoglobin, transfer RNA, and antibody. And now we're getting into molecules like glucose and methionine and carbon atoms. Ooh. Yes. So just to give you an idea of how the sizes of things relate to each other. So we've seen all the different sizes of things that we could possibly be looking at, and we have a variety of instruments that we can use to see that. One of the concerns that we have when we're trying to make an image is the resolution of it. We want to make sure that we can distinguish things from other things. All right, so resolution, being able to distinguish, see the distance between 2 objects and see them as 2 distinct objects. For the human eye, the limit is about 0.1 millimeters. Dealing with instruments that let us look at smaller distances than that, in the 1870s, the physicist Abe determined this resolution equation. Whoops, and that's not the right one. There we go. This is not on the test, but I felt I had a duty to include the actual equation. The shorthand is that the resolution of your image is going to be about half the wavelength of the energy that you use. So whatever wavelength of light that you're using, the ability to resolve objects is going to be half that wavelength. We can look at stuff with the almost naked eye. We've got some stereoscopes that you can just stick a plant under there and start looking at things. This is from a barley leaf. We have this very complicated structure here where the leaf is emerging. Fortunately, one of the things that we have at our facility are ways to cheat physics. We have software that can manipulate the images in a good way, taking what's in focus and using an algorithm to make sure that the picture is only made up of things that are in focus. So this picture was actually taken from a series of maybe 5 pictures, and then the software was used to squish it down and get everything in focus so that we can see the differences in the structure. The researcher was interested in trying to see differences in the cell shapes at different parts of the leaf. So it was important to make sure that everything was in focus to see that. All right, this is a cross-section of brain. I'm not sure what the staining, whoops, there we go. Not quite sure what the staining was, but we use staining to make things more visible. Apart from maybe blood or chloroplasts or things that already have pigments in them, biological materials don't have a lot of contrast, don't have a lot of color, so we need to use colorful stains to make things more visible. And some of those stains are attracted to certain molecules, ones that will stain lignin, ones that will stain nuclear material, ones that will stain fats. So that can help give us a much better image when we're looking at them under the light microscope. Example, oh, yes?
Can you talk a little bit about the issue of concussions? And there's something about concussions that they can only, if there's been some brain damage, they can only detect that after the individual has died.
Unfortunately, that's outside my expertise, so I'm not able to help you with that. I apologize.
Into my head when I saw it.
No pun intended, right? But nope. So the next couple images, these are fungal spores on cleared leaf tissue. We don't want those pesky cytoplasm getting in the way when we're trying to look at our cool fungal spores. So we frequently use chemicals to wash that stuff away. Then we can stain the fungi and see where they are. So we've got this spore, and then focusing down into it, we can see this structure, which has actually penetrated the plant cell below it. When I'm taking these pictures, we didn't have any of the fancy software, so I just had to take the pictures independently. But it shows you the importance of having things in focus. It would be a much more impressive picture if I showed you everything in focus, but oh well. All right, so those pictures were using natural light or white light. One of the powerful tools that we have available to us is using fluorescence. And this is not just, that fad in the 80s where we had all the great fluorescent clothing, but there are molecules that when you shine light at them, when they absorb a certain wavelength of energy, they will emit a different wavelength of energy. But the molecules, when they're at their ground state, they're just hanging out here, all right, minding their own business. Then they get a shot of energy because you're shining a light on them. So this energy state shoots up to here, to the next level. Then as that energy dissipates, it wants to get back to its resting state. It can't stay up here forever. So as that energy level goes down, there's light that's emitted. So this is the visual aid part. Everybody who I gave glow sticks to, get ready. All right. So the idea being that if you shine the light at the particular molecule, you're going to get a light that has lower energy coming out. It's not going to be the same light. So I've got my blue light here. All right. Where are my green lights at? All right, great. Everybody take a look around. If you all are the sample, and I am the microscope shooting a blue wavelength at you, you're giving me back green light, which has less energy. All right, great, thank you. Go ahead and turn those off. Now, if I was shining green light at you, take a look, we get red light back. All right, if I shine green light at you and you're fluorescent, you're not going to give me green light back. All right, great, thank you. I need those back afterwards. So we have microscopes that can detect that fluorescence. They can purposefully emit a wavelength, a specific wavelength of light, and then we can look for the different wavelength of light that's going to come back. Right, so this is an alga. This is Volvox. Now this has its own autofluorescence. We didn't have to add a stainer or anything to it. We shone a light, probably a green light at it, and we get red back. Right, but it's very cool. Volvox are very round and globe-like. These are HeLa cells. These have a couple of different fluorescent stains on them. The round structures here are the nuclei. The fibers here that are kind of bluish and some of the that are red are actin and microtubules. So to give you an idea of the cytoskeleton that's keeping the cell in shape. So we can use multiple fluorescent labels on one sample and then collect each bit of light separately and combine them into one image.
Gail, what was that thing? What was that last thing that what is that?
Those are cancer cells. HeLa cells, the Henrietta Lacks kind of lineage. I know that there are researchers that culture them on prepared glass or plastic, so they can grow on a single solid layer, single layer of cells. All right, so with the fluorescence microscopy, we can see all sorts of great information, make it look like terrific colors. There's another technique in microscopy that we can use to have even more powerful, convincing information, and that's called confocal microscopy. So remember the two pictures that I showed you, the fungal spore on the leaf, and I had to take two different pictures to be able to show you what was in focus? That's because there was stuff below the fungal spore that's kind of getting in the way, making my picture blurry. For that structure that was below the plant cells, all that plant material above it, making it unclear. With confocal microscopy, what's great about that is that only the light that's in focus reaches the camera or your eye or the recorder. So anything that's fuzzy above, anything that's fuzzy below, that's excluded. So this diagram helps to show that if we've got our sample here and the light that's coming from above where we want it or below where we want it, if it doesn't hit that sweet spot, you don't see it. Yes, somebody said clever. I totally agree. All right, so but it's a very powerful technique in that you can take pictures, you can focus through your sample, taking a picture as you go up through the sample, collect all those pictures, again, use a computer, smush them down, and get a very in-focus image. So that's one great thing about it. Another thing is you can actually virtually section your sample. If you were to take pictures throughout your sample from the bottom to the top, and you know about how thick the depth of field is, you can treat those as virtual sections. And then you create a stack of images and it becomes a volume, and I'll have a picture of that in a second. Those are the cool rotating, you've seen usually see with molecular models, and the molecule is just spinning around. We can do that with actual images captured on the confocal scope. Now, with the fluorescence that I mentioned, we shoot one wavelength, one color in, another color comes out. How do we not get those colors mixed up together? And here's where I can honestly say, it's all mirrors. No smoke, but there's mirrors. So we've got the source of our light, and it hits a dichroic mirror. Now, a dichroic mirror, it's kind of like, it's not quite a one-way mirror, it's like a 0.5-way mirror, in that it will reflect certain colors. If a green light is shining on it, for this one, green light's going to be reflected. So the green light hits the mirror, comes down, goes through the lens, gets to our focal plane. Then remember, we get a different color coming back, we're going to get a red color. So the red color is coming back up through the lens, hits the mirror, but red can go through. Red's not going to be reflected. So that gets to continue on its path. And again, confocal, hitting the sweet spot. Then it gets to our detector. So that's how we can get these optical sections so clearly and with multiple labels. So this is an example, again, fungal spore, but different one. This is one of those that's been squished. So this is where the spore is, and the hypha has started emerging, and now it's starting to sink its metaphorical teeth into the plant tissue to suck out nutrients. All right, but this is a whole bunch of pictures together because there's so much terrain on a plant leaf. All right, there's lots of bumps, okay? It's not going to be just a smooth, straight shot. So different things are going to be in different focal planes. But with the confocal microscopy, we can get it all in focus. With a fluorescent label that's specific to fungi and having the plant excluded, we get to see just the fungus without any of that pesky plant material getting in the way. All right. Pretty sweet. So this is a nerve. There's the blue part is the sheath, and then the yellow parts inside are actually the nerves. So that's a stack of images composed into a volume, and now we can rotate it. We can look around. We can look into some of the nooks and crannies.
Well, what is the depth of field that you can achieve with that? What did we call that microscope?
A confocal.
Confocal microscope. So what is the depth of field then, going through that, and what's the resolution in the XY plane?
For this one, I'm not sure. I know we can get down into below microns. So pretty small. We're working on about half a micron. Thank you. My ringer in the audience is helping me out with that one. So yes, about half a micron. And we've got, depending on the kind of microscope you have, some are better than others. Some have more powerful lasers. Also depends on how you prepare your material. So that can help with what the depth of field is.
Do you have a question right here?
So how dense can the material be to be used with a microscope like that?
Ideally, not dense at all. We really want it to, a lot of things can get in the way. There are many clearing protocols. Like I said, with the leaf, we tried to get all the chloroplasts and cytoplasm out. For animal tissue, trying to use things to keep all the structure in place, but get the lipids out, get the cytoplasm out there. If we can clear that, then we get a better depth penetration. All right, one more impressive 3D rotation. This is also nerves. And then it goes to side to side.
Sorry, is it correct to say then you have almost the same resolution kind of in the XY plane as you do in depth?
The question is, do we have the same resolution in the XY plane as we do in depth? No. No, unfortunately, we do not. It can become very visible when you take that stack. And if you've got it in the XY plane, it's like, yeah, everything looks great. And then you start looking at it from the side, and it's like, ugh, that's not so good. I actually took out a picture from my presentation because it didn't look so good because the angle I wanted was looking at the Z-axis. And it did not look good. I'm making a face for the people listening.
That was a good face.
Thank you. Thank you.
Okay.
Yes.
So we're looking at nerves in that last picture. Nerves of what? And what is, I mean, can you, whose nerves are
Whose
they?
nerves
Are we
are
looking
they?
at a rat's brain, or are we looking at a person's, like,
I believe.
what are we looking at?
What are we looking at? I believe
It's
we're looking.
impressive, but
I believe that's from a grasshopper.
What?
What? Grasshopper, yes. I don't know. Unfortunately, I did not take that picture. I have generous colleagues loaning me pretty things. All right, so with light microscopy, I mentioned that the resolution that we can get is related to the wavelength of the energy that we're using. All right, so if we wanted to look at the smallest things possible with a light microscope, we'd want to use blue light that's got the smallest wavelength. About the maximum resolution for that is 0.2 microns or 200 nanometers. And at the time before or earlier in the century, we really couldn't get beyond that, even if we had the best lenses and the best samples. So what should we do? Let's use something that's not light. Let's use something with a smaller wavelength. Enter the electron. So in the 1920s, two independent researchers came across some discoveries that both contributed to the development of the electron microscope. First, the idea that electrons can travel in waves. They have wavelengths. All right, so that's a potential energy source. The other idea being that magnetic fields can refract electrons. Now, that's also important. We've got, in a regular light microscope, the way that we alter the path of that light is through lenses, glass or plastic, but we're bending it to our will. All right, plastic and glass is not going to work on electrons. Electrons are just going to go through, or they're going to, go off and do whatever. But magnetic fields can act as lenses for the electrons, and that's how we can control their path. All right, so a reminder, the wavelength, distance between peaks. Within the electromagnetic spectrum, we have a nice little graph here showing radio waves with very long wavelengths. These would be very poor microscopes. We've got microwaves, and yes, the same kind that heat your burritos up. We've got infrared and ultraviolet sandwiching the visible wavelengths here. And then even as we go smaller, we've got various soft X-rays, hard X-rays, and then gamma rays, which, as everybody should know, can make small things big. All right, so a few more numbers, but the main point here, figuring out the wavelength of electrons. So how can we find out how small we're going to be able to see? If we have this, ultimately, at a certain energy, the wavelength's going to be of electrons are going to be about 0.4 nanometers. So our resolution, theoretically, is about 0.02 nanometers. So we can see some pretty small things. All right. Now, you seem to be a pretty savvy group. By a show of hands, how many of you have knowingly seen a picture from an electron microscope? All right, that's over half of you. Awesome. All right. For those of you that haven't, you probably have. There we go. All right. Those commercials, those allergy ads that show pollen grains the size of basketballs, those extermination pictures that show you ants the size of Godzilla. Those are likely done with an electron microscope. This up here I found on the web, but this is Gillette using a specific kind of electron microscope to actually etch a message into the blade of one of their razors. It's pretty cool. But yes, ants, bugs, they're great under a microscope. So types of electron microscopy I'm going to talk about tonight. We've got the transmission electron microscope, where you've got the sample is cut into very thin slices and electrons pass through it. This is much more similar to if you were in high school biology and they made you mount that piece of onion skin onto the glass slide, or you got the Carolina biological slides of the lichen cross-sections. You have a very thin slice, the energy is passing through it, the energy that makes it through makes your image. The resolution, theoretically, is about 0.2 nanometers. Usually it's about 1 to 2 nanometers for biological samples with a really great microscope. The second type of electron microscopy is a scanning electron microscope. This is just going to show you the surface of your samples. The electron beam hits the sample, and electrons are emitted from the sample, and that's what makes our image. The resolution is about 3 nanometers, again, theoretically. In practice, with a good microscope, 5 to 10 nanometers. This pretty much repeats what I just said, but now we have pictures. We have a nice, we have a nice picture of a transmission electron microscope. They tend to be bigger and taller. Both of our electron microscopes, we have one of each in the St. Paul facility, take up the room of a generous walk-in closet. So we've got the transmission electron microscope here. This is actually our scanning electron microscope on the St. Paul campus. This is a former colleague of mine operating it. So you need some room. The transmission electron microscope is, the design of it is very similar to a light microscope, except it's upside down. Well, and it's much larger. But in terms of where the energy is coming from and where the final image is made, it's kind of reversed from that. Diagram here, the TEM is right side up, because we're all about EM right now, so light's going to have to change for us. So for this light microscope, we have the light source at the top, because on the TEM, that's where we have our electron source. That's where the beam comes from. The beam travels down, goes through lenses, goes through the specimen, and then more lenses. And then for a light microscope, we're going to image that directly, or we're going to, on the TEM, we're going to see those electrons hit a screen or a detector and form our image. So again, we've got the energy going through our sample. And note the lens similarity here. Here we've got glass or plastic for our lenses for the light. These are coils of wire. These are electromagnets. That's how we're changing the path of the electron beam. All right, this is all under vacuum. We need to get all the gas and dust out of there so that our electrons can travel freely. If we've got gas or particles in there, it scatters our beam and makes it less distinct. We get a poorer image. It's like turning your high beams on in the fog. We don't want that beam to scatter. All right. The anode drawing the electrons down from the source, the magnetic coils to focus things, and then we get our image. Now it's great just sticking a sample under the microscope, but there is a lot of preparation that goes into making a sample good for the TEM. I will try to be brief, because goodness knows the protocol's not. We have samples that we fix usually with chemicals. That's what we call conventional fixing. We've got glutaraldehyde, paraformaldehyde, some nastier stuff later on. But we wanted to fix them with chemicals to create cross-links chemically to make sure everything stays the way it's supposed to be. We then embed it in resin so that we can take the really thin slices. We've got it's such an old picture. This is an old-timey microtome to be able to cut the thin sections. It's like a very small, expensive deli meat slicer, pretty much. So once we've got the slices cut, we collect them on a grid. This is about 3 millimeters in diameter. Super teeny tiny. When I cut a slice for the TEM, when I'm cutting these slices, it is less than 1 millimeter wide at the bottom. The thickness is even tinier. We cut generally between 50 and 100 nanometer thick sections. We do that with a diamond knife. so that we can get that thinness. That's about 1/200th the thickness of a cell. So it can take us a while to get through a cell to visualize it. So once all of this is done, then we get our image. We've got a thin section here. This is from a liver cell. This is a nucleus. You can see the nuclear membrane here. We've got some ER around it. These round things are mitochondria. So how do we get that beautiful black and white image? Biological material really is easy for electrons to pass through. If we didn't stain or treat our samples, it would just be very low contrast, blah. So we use heavy metals. We can't use colored stains. We can't use any of the we can't use the fluorescent stains that I just mentioned earlier, because those all involve light and color, and there's no color here. If you see an electron micrograph with color, somebody has added that. Okay? I've seen plenty of TV shows where they have the point of view down the microscope, and it's some electron micrograph that they've colored prettily. Don't believe it. All right. Okay. So we can't use colored stains, so instead we use heavy metals. The heavy metals that we use include lead, uranium, depleted uranium, and tungsten. These large atomic molecules that if an electron ran into it, the electron would just bounce off. It's like a BB hit in a basketball. Those electrons are knocked off their path, so they don't get to pass through. So that ends up being a dark area in the sample. So this is neuroblastoma. We've got nuclei again. They've got a lot of stain around the membranes here. And this is likely heterochromatin, some nuclear material that's more condensed, so it's picking up more stain, so it's darker, because electrons are having a tough time passing through that. For nostalgia's sake, here's a TEM micrograph that looks like something you've seen in a biology textbook. In fact, because it is. We've got our plant cell wall, the big square here. In the center, we have our nucleus. We can see the nucleolus. Let's see, where are they? We've got some mitochondria in here. Mitochondria look like small little jelly beans that have worms in them. If you look at them under a TEM, that's how they're visually distinctive. We've got endoplasmic reticulum, these sheets here. Ribosomes, very small dots. But just for reference, a little plant cell there that actually matches the model of what you've seen in textbooks. I've mentioned fluorescence microscopy and transmission electron microscopy. And one of the things that we endeavor to do at the imaging centers is using more than one method to try and get the answer that we need, the data that you're looking for. Sometimes one method's not going to cut it. Sometimes using two methods in tandem, two microscopes together, can give us the information that we need. This is some work from a client who has looking at a fungus. Yay. This is a fungus that produces a toxin when it grows on a commercially important plant. And they're looking at how this toxin is made and where is it being made. So they've got some, these are some specialized light micrographs. The fungus will grow, will create the toxin only when it has a specific, it's growing in a specific medium. All right, so this is non-poisonous. This is no toxin produced. And here, it's growing in the medium where toxin is produced. So we've got our little light pictures so we can see where the cells are. The enzymes that help make the toxin have been labeled with a red fluorescent protein. So here we can see, you may be able to see the little red dots, but there's none up here. Then you can put these images on top of each other and see where those structures are in the cells. And that's just at the light level. What's great, for this project, they looked at both light microscopy and transmission electron microscopy. We fixed samples, we cut slices, and we found matching features. All right, here on the left, we have a strand of the fungus, and it has these structures. These green parts, those are nuclei. But you can see that the red, where the toxin's being produced, there's kind of this kind of half moon shape around some of these nuclei. And then we saw that in the micrograph. Yes, it's colored, I know, after what I just told you. It is okay to color these things sometimes to try and help the audience understand. But this is the nucleus in green, just like it was green in the light microscope. This is the endoplasmic reticulum. And in these fungi that are making the poison, that are making the toxin, we tend to see a lot more of this. And they're just hugging that nucleus. They're just right up close to it. Another feature, we see some of these red dots in the light micrograph in the fluorescence picture that are just red. So they're not near any nuclei. And we saw that with these structures, these whorls, and it's just membrane on membrane on membrane. All right, all containing the toxin-producing enzyme. Question?
And you're trying to produce disease-resistant plants.
The question is, are we trying to produce disease-resistant plants? Likely eventually. This is more the basic. They're trying to understand the biology of the fungus itself. So trying to see where the poison is being made, where the toxin is being produced. If they can break up that pathway, then yes, potentially they can mess up the disease-causing features of the fungus. One of the projects that I worked on involving transmission electron microscopy was the fungal tree of life. If you have a connection to the museum, I believe you've heard of the tree of life in terms of, yeah, Leah's laughing. The idea is trying to see how everything, what's related to what, is the short version. And I worked on the fungus version of that, seeing how the fungi were related to each other. Now, fungi have a reputation, like lots of other things that have been classified and named, based on things that we can see with the naked eye. All right, what does the mushroom cap look like? What does the color look like? Oh, they must be related. This is a diagram showing many different kinds of fungi and the fungal groups. The take-home message are, these are the types of fruiting bodies, whether you've got an umbrella cap, whether you have gills, whether you have spines, whether you have pores. All right, there's more than one for each group, each family group of fungi. That's not a very reliable indicator of what's related. So generally not going by that so much anymore, but with the advent of all the gene sequencing and comparing DNA, that's a very powerful tool, very informative. Not the only thing, though. This particular grant was made to be able to incorporate both DNA, both sequential data, and also ultrastructural data. Again, people were like, oh, we don't want to do anything. We don't want to see how they're related based on what things look like. Goodness forbid. All right, but these subcellular structures can be very informative. Some of these subcellular structures, many genes need to be working to be able to make them. Lots of features are common to all groups. All fungi have a nucleus. All right, if we can find something with a trait of the nucleus that's different between groups of fungi, that can be very informative. Also, there's different selection pressures. We're not necessarily going to, depending on what host you're on or who's going to eat you, that's not necessarily going to change how your nucleus divides. All right, so these traits can be very informative in seeing how things are related. The structures that we looked at the septa. This is, imagine fungus being just all plumbing. It's all tubes, and there are parts where it's walled off where the cell happens, but there's a hole in that wall so that you can still have the cytoplasm running back and forth. All right, fungus is just tubes. But at those cross walls, there's many different structures. And those structures seem to be similar between different fungal families. This is an example. These are drawings from ultramicrographs. These are the rust fungi that often produce that, literally, a rust-colored spore. We've got smuts. I know it when I see it. And then we've got mushrooms. I put that in quotes. What we think of as mushrooms with a cap, and it's growing in the ground, and we put it in our salad. All right? So you can see the similarities between these two, or these three, within the three groups. All right, so that can be informative. All right, also how a nucleus divides can be informative. This isn't sequential. These are different options. If you thought a nucleus divides a certain way because of your biology textbook, that textbook lied. All right, or was simplified. You may have the membrane enclose the entire nucleus. You may have the membrane break apart at the ends. You may have it disappear altogether. Those traits can be also conserved within fungi. We looked at a particular fungus, Auriscalpium vulgare. You'll notice it does not have gills. It's got these spikes. Auriscalpium, it's the ear pick mushroom. Yeah. But we had it in culture. It is a little studied mushroom, so we were breaking new ground with this. Nobody had studied it at the ultrastructural level until this. So we got to see the septum. This is the cross wall. Here's the hole, and it's got this cap on top of it. It's like someone took a wiffle ball and cut it in half and smushed the edges. This is all one thing, but it's got holes in it. This is kind of a glancing cut of the cap, where you can see the wiffle ball texture. And you can see how it spreads all the way down there, which is unusual for fungi. We also got to see, I'm not sure if it's visible, but there is a layeredness to the cap. And we were able to see that with transmission electron microscopy. Also with cell division, this is a fungus spore, still attached off screen. But we've got the spore, we've got this clear part that's wavy, that's the cell wall. In here, here's the nucleus, and it's dividing. And we can see the nuclear membrane is mostly intact. We've got the spindles here, the microtubules that are attached to chromosomes and pulling them apart. And then we have the organizing structures for those spindles here. So that was not known before, and we got to see it. So the microscopic features can be important in figuring out how things are related. Switching gears to the scanning electron microscope. Oh, sure.
It sounds to me like, sorry, what was that microscope that we were talking about before?
The microscope we were talking about was the transmission electron microscope.
So it sounds like the transmission electron microscope does to things on the cellular level what a CT scan does on a body level.
It can, yes.
Okay, thank you.
Yes. Mm-hmm. So switching over to the other type of electron microscopy, this is our scanning electron microscope. This is my brother. He was visiting some years back, and he is practicing safe science. He is in a lab coat. He has eye protection. He has a thermal glove on, just the one. And he is not looking at anything. So I am, as a sibling, I am morally obligated to include that picture. So like I mentioned with the transmission electron microscope, we have thin slices, the electron beam goes through it. With the scanning electron microscope, we have an electron beam, it hits the sample, and all we see is the surface. We've got, similarly, an electron gun up here, it forms a beam. We have our magnetic coils to focus. The beam hits the sample, and as the beam literally scans back and forth over the sample, and there's a detector within the chamber, and so as the beam moves from point to point, the detector detects how many electrons are being emitted from the sample. If there are a lot of electrons that are coming off, that's a bright spot. If there's not, that's a dark spot. So as it remembers the position each time, it forms the image. Sample prep for the scanning electron microscope, similarly involved. It can be a little easier though. We do still have the chemical fixation. We've got the tissue dehydrated with ethanol, but then we completely dry it. We don't put it in plastic. We want to make sure there's no water whatsoever in our sample. This is also operated under a vacuum. If we have water vapor in there, it gets the insides all icky and gets water vapor inside, and again, that can risk scattering our electron beam. The samples are mounted on special stubs, and we coat them with a thin layer of metal. The image is formed, like I said, by the electrons coming off the sample. Biological material doesn't like to give up electrons. It's kind of selfish that way. But the thin layer of metal, if we coat our samples with that, metal loves to give up electrons. So if we have that metal coating, it improves the quality of our signal, of the amount of electrons that are coming off, and we get a better picture. Oh, this is the classy dryer that we have. It's similar to ours with the orange and the wood paneling. Yes. When the beam hits the sample itself gives off electrons from a certain level, and it gives off other energies that we're not going to talk about here, but it's not just electrons that are coming off. There are other electrons with different energy. There's also X-rays that are being given off. So a variety of energy is being released by their sample once you're hitting it with a high energy beam. When we see the pollen grains, when we see the ant Godzilla, those pictures are generally made from the secondary electrons that come off, that are emitted from the sample. There are cornucopia of images here. Our red blood cells, 8 microns. We've got an algal cell that has bacteria on it. So it's like a giant soccer ball that's got gummy worms all over it. Up here, this is going to blow your mind because it blew mine. This is a part of a crab stomach. Right. Inside a crab, it's got these two structures that grind against each other to basically kind of chew the food and make it so it can digest it. So this is one side of that. So it's very textured and it grinds. These are human neuroblastal cells growing on a surface. So it's not so much like a tissue, but they're growing in culture. You can see the stretchy shape and some of the structures that are coming off from that, touching each other. This is one of my favorite pictures. I took this last winter. This was on my bucket list, to look at snowflakes under the scanning electron microscope. This involved having a cold stage. Doesn't have to, but our particular SEM has a cold stage that you can lower the temperature down to minus 130 degrees Celsius. So we can keep the samples chilled, because again, high energy beam hitting your sample. But it being frozen and coated with metal, we get to see these flakes. It was a good snowstorm going on there. So they're kind of bundled up together, but we've got different kinds. I mean, and they're different, like the adage says, but different ages. There was a young one back here, but you can see the differences in the flakes. One of our users is looking at mutants and normal wild type, barley plants. So with the plants, there's a waxy coating, the cuticle, on the surface of plants to protect it from water, protect it from insects, protect it from fungi. This is the surface of one of those leaves. It looks a little deflated because these samples were just dried. Since this person was only interested in the wax coating, we didn't have to go through all the complicated chemicals that might risk disturbing the wax layer. So we just dried these in an oven and coated them with metal. But you can see the cell walls, the outlines of the cell walls. Here's a stomate right here. This fuzziness is the wax. And if we look up close, we see this angular plated crystalline wax there. So that's quote unquote normal. They also have what they call glossy mutants. So the waxes are not as pronounced on the sample. I'm not sure what the round structures are, but the amount of wax, all those flakes, is greatly reduced. And here's a close-up of that. Just little bit here and there. This part of the leaf has virtually none. Here's a stomate. Here's a little beginning of a plant hair, cell walls. Then we looked at a different structure on the plant that was a regular normal wild-type plant. And then we saw something unexpected. This was the wax that we saw. We're not expecting this. It's almost like cotton candy. This is a side view of the leaf tissue. This is only 25 times original size. All right. The leaf is kind of curled up on itself, but we can still see that surface. And if we go closer, we can see the fuzziness and how much that wax layer sticks up. When you look at it with the naked eye, that was actually one of the challenges of preparing these samples. If I even touched it, if I looked at it funny, I would wipe off that wax. So it was a very delicate procedure, just trying to cut it and grab it from the edges and place it without disturbing that layer. And you can see why it'd be so challenging, because it's so airy and delicate. So that was all SEM. Another project, and this is more my graduate work, we worked on a fungal pathogen on poinsettias. Christmas is pretty much ruined for me, because if I ever go past a big display of poinsettias, I don't see the beauty of the poinsettias. I'm immediately wanting to get closer and say, okay, how many colonies are there? Where are all the white spots? I'm scarred. But this particular fungus only grows on the surface of the plant. It occasionally will penetrate the top layer of the plant and suck nutrients out of it, but most everything else is just on the surface. And it produces its spores on the surface, which is why it's called a powdery mildew. This hit the U.S. in the early 90s, and it was quite a problem for growers because A, it was new, B, they didn't really have any ways to fight it, and C, because you're dealing with a very visual plant and it's sold based on appearance, if you have any blemishes on it, you can't sell them. Although there were some enterprising growers who tried to sell their infected plants by sprinkling glitter over this and calling them snow poinsettias. That is a marketer's dream and a pathologist's nightmare. So I was involved in some of the basic research, trying to figure out what were the best environments, what were the optimal conditions for spores to germinate. So we got some great SEM pictures here. These are taken at different time points, 2, 6, 12, 24, 36, and 48 hours after inoculation. After you put the spores down, then what happens? We've got the spore here. It's producing just a little bit of a germ tube, and then that expands into this structure. That's where the magic's going to happen. That's where it's going to start going in and trying to get into the plant. If it fails, it'll have to make another germ tube. And then eventually, we've got longer hyphae, longer strands, more plumbing going on. These are from more established colonies. You can see the hyphae growing over each other. Here's the spore-producing structures. What was really interesting about this fungus that hadn't been reported previously, the base cell here has this little crook in it. A lot of the fungi in this particular family don't have that. So we were hoping, oh, it's got this bent cell. We should be able to identify it. That'd be great. No, but it looks really cool. It's a very good identifying feature. And here's part of a colony. You can see where the spore started here, and it just starts sprouting, going off in all directions. All right. I didn't realize I had that in the other picture, but that's a nice close-up. So I did research on that, and I went to a different lab, kept working on the same fungus, but wanted to see it in a different way. We wanted to see what was going on in the plant. So we used TEM for this. Here's on the surface, I showed you the structure there. That's where it's going to penetrate, and then it's going to make this structure, the haustorium, inside the plant cell and start sucking out nutrients. So how can we see that with the TEM? Like so. So these are cross sections. Here's the epidermal layer of the plant. There's a layer of flat cells. That's where the fungus is going. It's not going anywhere further than that. It just wants to set up shop and start sucking the life out of this plant. Here's a close-up of that. There's all these lobes. It's like if you had a giant balloon and you stuck your hand, and it was blown up, and you stuck your hand into the balloon, your hand is like that fungal structure sucking the nutrients out. It doesn't pop the balloon. It doesn't pop the cell membrane of this cell, because if it does, that's going to kill the cell. This fungus is an obligate parasite. It needs the plant to live. So it wants to be gentle when it's doing this, but it still needs the nutrients. So these lobes, or like if your fingers were those lobes, it increases the surface area for them to be able to absorb the nutrients. We've got close-ups of the neck here, where the penetration peg came down, and different traits along there. So you're able to see what was going on with this fungus using scanning electron microscopy to see when things were happening, and that gave us the information so that we'd know how old our colony should be before we start taking transmission electron pictures of them to see how the fungus was getting inside. And with that, those are some examples of the kinds of tiny worlds that are visible, how we can use different instruments to see those worlds and collect data on the living world around us. I'd like to thank my fellow UIC staff members, especially Mark Sanders, Grant Barthel, and Guillermo Marquez for lending me some pretty pictures and for their advice. The AFTAL project, where we got the how fungi are related in the Dave McLaughlin lab. Marka Banish and Corby Kistler, who gave you the cool light up nuclei and ER with the poisonous fungus. Ron and Zenith had the barley projects, and then the poinsettia projects were at Michigan State University and University of Georgia. So with that, I'd be happy to answer questions, and thank you very much for your attention. I appreciate it.
Thank you so much. I'm going to start with questions right over here.
So what's the future? Like, we have light, electrons. What's the next?
According to the website that had that world's biggest microscope, neutrons are up and coming. Also, there's also a field that's trying to cheat physics in terms of trying to get better resolution from light than that equation says we should have. And it involves how how the specimens are prepared, trying to get them as clear as possible. It involves what kind of energy that you're shooting at the sample. A lot of what you saw there, it's one beam and it's one wavelength that's hitting there. And if that energy is too much for it, you risk ruining your sample or killing your bacterium or something. So what if you had, instead of one high energy beam, what if you had two lower energy beams hitting it and meeting at the middle? And what could you see from that? So that's some areas that are being worked on.
So on the SCM images like this, we see a lot of depth. So if the laser or if the electron beam is just scanning across it, where does the depth information come from?
I'm not sure. The way that the samples are visualized in the scanning electron microscope, if you're not getting a strong signal from an area, if an area is not releasing a lot of good electrons, it looks dark. So if you've got a good sample and it's giving you a good signal, then you get a good image. You can adjust or influence the depth of field with the scanning electron microscope. It's almost the same as a camera. Depending on how far away your sample is from where the beam is coming out, if you have the sample that's further away, you can increase that depth of field. However, you also risk getting a crappier picture because your sample, and when I say crappy, that's not scientific, but in terms of if your electrons have to travel farther to hit your sample, they risk going off path. And then if your emitted electrons have to travel farther to get to the detector, they may not make it. So you may get a fuzzier image if it's farther away, but you have great depth of field. If you move your sample up closer to where the beam comes out, your beam is right there, and those electrons are right close to the detector, and it's great. However, you have a shallower depth of field, and you also risk possibly messing up your sample because high energy shooting at it can't, depending on the specimen interactions, it can alter your sample. So you have to be careful.
I was also wondering, all these great pictures and a lot about fungus. So is that the area of research that you and your colleagues have? And is it split between you guys doing your own research and just being asked to take images or contracted to take images for other parts of the U or for private companies?
It's very difficult to have one's own research in this particular role. Our job is to help other people with it, and that's the duty. There are occasionally side projects that we can add on. Maybe add an extra experiment to somebody else's project. It's not so much that we have our own independent research. A lot of this stuff was from my graduate work, and I'm okay with having it out there. So I got the permission to be able to present it. So fungi is my background, and fungi also have cool pictures. But I'm currently employed as a microscopist, and that is my pun intended focus. So I don't get to do a lot of exploring myself. More with techniques. We try to improve the techniques that we use to preserve and prepare samples. So that is one area where we can make new discoveries.
It's two. Maybe they're kind of related. Maybe. So when you stain things, you use these heavy metals. So how and why? Well, how do they associate with certain structures, and is that a uniform thing, or is that why you have all kinds of stains? And just, if you have any nerdy details on that. And then, because how does it make sense that it knows where to go or where to stick? Anyway, and the other one is, it sounds like when you coat it with metal, it's kind of like plating, so why does the metal stick to your sample?
Physics. For the first question with using the heavy metals, Some of the heavy metals and their salts are attracted to lipids, are attracted to fats, are attracted to proteins. They can be pretty general, but sometimes you want to give it an extra boost and have something that's more just widely staining in general to help visualize the structure you're looking for. Sometimes you put in something specific that, like for membranes, I know in the past permanganate salts have been used for that. So there is some specificity. Also, pH can sometimes play a role. So yes, although quite often it's a standard stain. And then if it doesn't work, then we take a closer look at it and say, okay, how can we improve that? And then your second question was, how does the metal stay on? Hope. It's a very thin layer of metal. It's nanometers thick. So the way it's applied is you have a sputter coater. You have a device that you put in. You put your sample in. It's under vacuum. In the same space, you have you can have a magnetron and an inert gas. And then as you charge that gas, that plasma just hits all over the place. It hits the metal target. And then the metal that comes off of that target coats everything inside, as I can testify because I have to clean the thing.
The metal atoms are associating with each other as opposed to the sample, right? They're like bonding each other to form this surface.
Maybe. I am totally blanking on how a sputter coater works, unfortunately. Sorry. But yeah, it's a very thin layer. The metal sticks on there. It's not like dunking it in or anything.
Beautiful pictures, by the way.
Thank you very much.
I was intrigued by your talking about how you prepare the specimens. I've worked in a hospital lab dealing with human specimens, and we always used wax. Have you ever used wax, or have you always used resin because of the small size of your specimens?
We use resin because of the small size. Our lab is not necessarily set up for that scale. I know wax is used for larger scale things, but we are pretty much ultrastructure, so it's resin all the way.
Your fascinating demonstration here makes me wonder if this is the future of the traditional MRIs, or did you get influenced by the MRIs, or are they both separate entities?
They're both separate entities, but I can see how information from one can help inform the choices you might make to use the other.
Hopelessly naive question, but when you shoot electrons, what is the source of it? How does that work exactly?
Where do the electrons come from? There are multiple electron sources that can be used. Our microscopes can use tungsten filament, and it is tungsten in the quote-unquote old-fashioned light bulbs. The tungsten is a wire. It's in a specific shape. It's in a V shape, and as you run a current through that wire, you get heat, but you also get electrons emitted, and the anode uses charge to direct the electrons down in that direction. And they go through apertures, and they're focused with magnetic lenses to get them to go where we want.
How long does it take the scanning electron microscope to complete one image, to complete a whole course, to make that sample? And also, are either the scanning or the transmission microscopes, would it be possible to image a sequence or an event on one sample, or is the process of preparation destructive to that?
Answer the second question first. We can go back and re-examine samples. I have samples that my predecessor prepared for class tours, and they still work. So they've got some longevity. The sample's not destroyed; it's not consumed in the visualization process. How long does it take to take a picture?
The scanning electron, how long does it take to complete one?
How long does it take to complete one scan? You actually get to change that. So as we're looking around a sample and trying to find where the cool places are, we can have a very rapid scan, like, in fractions of a second. Once we get, but it's very snowy. It's kind of staticky. There's noise in that image. It doesn't look so great. You wouldn't publish it. But if you, when we're ready to take a picture, we will set the scan speed to a slower speed. So over 30 seconds, the beam will scan the area and give us our higher resolution image that has more detail, less snow, less noise.
I was curious about as researchers come to you with their samples that they want you to prepare and scan and look at and photograph. How often do they understand the technology and the procedure that they're actually submitting their own work to? Is there a connection there, or are you really kind of the mysterious place where these things go and then they come back and they have what they wanted?
It's a range of informed users. So there are some people who say, I saw it in this paper, I want to do it to my organism. And you're like, okay, but they had a $2 million microscope. And sometimes a person has done microscopy themselves and said, I used to do this in grad school, and but we just need the one picture or the two samples done. So could you, we'd like you to do that. So some people come informed. I regularly suggest before I meet with potential users, if they have a paper that sparked their interest or shows what they're interested in, with the data that they want to get, I encourage them to send it to me or bring it before our meeting so I can take a look at what do they want, what are they expecting, what are they looking for, and how can we meet that need, whether it's what they saw in the paper or whether we say, you know what, you can really see the same thing if we do some fluorescent labeling and let me introduce you to my colleague.
It's like being recommended to bring your picture that you saw in the magazine to your stylist.
Yes, it's very much like that.
Excellent. I just had one last question, and of course we have time for a couple more from the audience. But I'm curious, because being at the Bell Museum, we do have our Explorer Dome, and we work with the astrophysics department to do viewing of the very large structures. And that's something that has been brought to the public in some ways through outreach. And are there ways to facilitate, public viewing of this kind of materials through this kind of technology that are going on, or is that something that you think about doing, or maybe it's already happening? I feel like there might be people here that would be interested to know.
Well, for stuff like this, Café Scientifique is a great avenue. Also, we do have people from our lab do participate in Microscope Day over at the Science Museum in St. Paul. The Minnesota Microscopy Society frequently works with that. I remember one year I helped, and actually a company brought one of those tabletop SEMs, not the basketball-sized kind, but the dorm fridge-sized kind, and we got to look at coral grains and fly heads, and students and kids and grown-ups got to see that in action. We also do tours for classes. We've had high school, undergrad, graduate courses, come through either a 5-minute tour, a 20-minute tour, or they make us part of the lab and they have to be there. So that's part of our outreach to try and make this more visible to people outside the university.
Excellent. Right up here.
How did you get interested in this field?
How did I get interested? I liked plants in high school, and when I got into grad school, that's when I was offered the opportunity to work on that new fungal disease. And we were trying to do what we could with the light microscope, and my advisor suggested, hey, maybe SEM might be helpful with this if there are structures there that we can't see with the light microscope. So I learned SEM for that, and then continuing on in grad school, had an advisor that was much more about the TEM and identifying certain carbohydrates and proteins in the plant-fungus interface. So then I got to learn TEM.
Thank you guys so much for coming tonight. Thank you to Dr. Gail Celio from University Imaging Center.
Thank you, everybody.
And please consider joining us. Oh, please return your light sticks to to Dr. Celio. Make sure to pay your server on the way out, and or before you leave, rather. And join us next month for the physics of confections. Thank you.